So in this video, I wanted to talk about how different muscles can contract. So first, let's talk about the myofibrils in skeletal muscle. So these myofibrils are going to be able to contract, and that will result in the entire muscle contracting, if they're all contracting, right? If all of these myofibrils are contracting, then you can imagine the entire muscle would contract. But how do they actually do that? Well, these myofibrils are going to look like this. Okay. Or like, let's just say it looks like this. And they're going to be they're going to consist of these different parts, these repeating parts. Okay. So this is one myofibril, and it consists of several of these parts. Okay. So right, myofibril. Okay, and each of these parts, each one, is called a sarcomere. Sarcomere. Okay, each one of these. Okay. And each of these sarcomeres is going to be able to contract on its own. So each of these sarcomeres is going to be able to contract, and then therefore the entire myofibril will contract when those sarcomeres contract. And this is a diagram of a sarcomere. Just say this. Really quick. Actually, I'll just draw straight lines. Okay. So just a really basic drawing, not exactly, not completely visually accurate, but it's just to give you an idea. And this would be a Z-band, and on either side of your sarcomere, you would have Z-bands. Okay, you'd have a Z-band. Okay. And between these two Z-bands, there are going to be two proteins. One is going to be called actin. Okay. They're going to be placed like this, and I'll explain how they work together to contract in a second. So this is actin. Okay. I don't know why I made that arrow. Okay. And you're also going to have myosin. Okay, and it's going to look like, well, I'm going to show you, just give you a little idea of what it's like. Okay. They're probably going to be more, but this is just to give you like a basic idea of what's happening here. On either side, okay. All right. So there's actin, and then there's myosin. Actually, then there's myosin. Oh, whoops. Okay, and there's myosin. All right, and then there's also titin. I should add, and explain what titin does in a sec. So titin, and titin is basically connected to the myosin and then connected to the Z-band. All right. Yeah. And then there would be several of these sort of rows. So there would be several of these. Okay, so we continue on this, and then there would be another one, this. Okay, just really quick, just to show that there are many. All right, and it just continues on, okay? And then there would be some number of these actin myosin parts, okay? Oh, and I should also mention that we label the different parts of this as, just to give it some perspective, um, uh, from the Z band to the beginning of the myosin heads, so these things right here, these myosin heads, and we'll talk about them in a sec, that's called your I bands. Those are called your I bands. And then between the first myosin head on the left and the first myosin head on the right, this right here, that is called your A band. Okay, so just to put it in a little, just to give it some perspective. Okay. All right, so that's just labeling it. But how does this actually work, right? Well, myosin and actin actually work together. So I'll zoom up. Say this is the right side. This is the right side, right? So this is what the myosin protein looks like, sort of. Not exactly, but this is just to give you an idea. And then your actin would be over here, okay? Let's just draw it as a line. And what can happen is myosin or ATP can be used, okay, so that myosin can move along or like push against this actin, okay? These myosin heads will push against this actin. So this is one myosin head. And each of these myosin heads will be able to use ATP to push against this actin, and that will make it so that the actin moves this way in relation to the myosin. And basically, you can imagine if that happens, if all of these myosin heads are doing the same thing, they're pushing against, or they're pushing against the actin, then what's going to happen is these myosin heads are going to move further apart from each other. So this myosin head is going to go this way. This myosin area is going to go this way. This myosin area is going to go this way. And what's going to happen to the actin is the actin is going to move towards itself. And what that is going to result in, right, is these two Z bands coming towards each other, okay? Because the myosin heads are sort of pulling the two actins closer to each other. So the Z bands are going to move towards each other as well. And then the entire sarcomere is going to contract, okay? But it's not exactly that simple, because we need some way to regulate this. We need some way to make sure that this only happens when we want it to happen. This only contracts when we want it to contract. So let's talk about how that's actually regulated. So each of these actins is going to have two proteins on it. One is going to be called tropomyosin. So I'll just do this. This is not really an accurate representation. This is really just to give you an idea of what's happening, like a basic idea. Tropomyosin. And tropomyosin is going to be blocking these myosin heads from even interacting with the actin. So they're not going to be able to push the actin in the way they want to make the sarcomeres contract and the entire muscle contract. When this tropomyosin is blocking these myosin heads. And then there's also going to be troponin scattered across your actin, right? And troponin it has the ability to actually move the tropomyosin when it is exposed to calcium. So when calcium is present, then it will move the tropomyosin so that the myosin heads can interact with the actin to make the muscle contract. So basically, these myofibrils contract when there is calcium. Okay. All right. So that is like the structure of each sarcomere and how myofibrils are able to contract and how muscles are able to contract, right? So now let's talk about how we regulate the calcium that interacts with this structure, right? Because we need some form of we need some way to regulate the calcium, right? Okay, so how that works is when you have a muscle, going past boundary. Okay, what's going to happen is well, actually, let's just start by drawing everything. Okay, so let's say you have some nerve here, okay, or your this is nerve, and it's going to nerve, and it's a nerve, and it's going to send a signal. Okay, it's going to send a signal to somehow interact with this muscle cell, right? This muscle cell here, right? Actually, basically, there are going to be a bunch of these nerves that are leading to your muscle, each muscle, and when they lead to it, they're going to reach muscle. Okay. All right. Okay. So let's just say this is muscle. Okay, and then it continues on. So this is your sarcolemma, like we talked about before. Each muscle fiber has a sarcolemma, which is its membrane. Sarcolemma. Okay. And it's going to have all of these little divots, I guess you could say. These little divots inside it. And then from a bird's eye view, so if you were looking at it like this was your eye, and you were looking at it like this, then it would just look like a bunch of little circles in it. Okay, like a bunch of little circles. And these, and there are going to be these divots, and there are also going to be sodium. Actually, I'm just going to say sodium pumps. As well as calcium pumps, calcium ion pumps, sodium ion pumps, calcium ion pumps. Okay, so I'll draw one here. And there are going to be more than this, but I'm just going to draw them. Calcium. I'm just going to draw one of each to keep things simple. Okay. So if you remember from the neuron videos, we said that neurons can transmit signals. Nerves can transmit signals using sodium and potassium gradients. So as the signal goes, sodium go in, and then so sodium will keep going in. And it's not alone. Calcium is also going to be going in as the signal goes along the nerve. And if you haven't watched the neuron videos already, I suggest you go watch those right now. But anyways, or finish this, or do whatever you want. Anyways, so the sodium comes in, like we talked about before, but we haven't talked about this, but calcium will also come in. In this case, it'll come in, and then calcium will be, basically we have these little vacuoles, okay? These little vacuoles, vacuole, I guess, and each of these vacuoles is going to contain acetylcholine, or acetyl choline. Okay. And these calcium ions are going to be able to bond, or these vacuoles have proteins that will grab onto these calcium ions. Okay, so now the calcium ions are attached to them, okay? Right? And that will make it so that these vacuoles are attracted, or they want to go to the terminal, to the end of this nerve. And what's going to happen then is the cell is going to fuse with this terminal, and then it's going to, first it's going to be like this, right? It's coming towards it, and then it's going to actually fuse to it, and then this is going to disappear, and then the I forgot to draw the calcium that's attached to it. Okay, this calcium attached to it. The acetylcholine from inside the vacuole is going to be able to leave through the terminal. All right, so this acetylcholine is now going to be in the synaptic cleft. Synaptic cleft. Okay. Yep. So what happens then is this acetylcholine. I guess I'll just draw it like this. It is going to. I guess I'll draw. It's going to cause these sodium channels to allow sodium in. Okay. And I only drew one, but there are lots. So there are going to be a bunch of sodium ions inside your cell now. Okay. Now these sodium ions, they have a charge, right? So that's going to result in a voltage gradient between the outside and the inside. So there are enough positively charged things inside here where there's a voltage gradient. And that voltage gradient is going to, I won't go into details of how, but it's going to result in the calcium channels opening, and it's going to allow the calcium. That's going to result in calcium coming in to the cell. Okay, so now I want to talk about something in our muscle cell that is called the sarcoplasmic reticulum. Sarcoplasmic, it's in your muscle cells. It's called the sarcoplasmic reticulum. Reticulum. And actually, I'm just going to move it actually. Okay, let's just move it because I want it to be closer to, I guess it'll cover the, okay, whatever. I'll put it right here. Make it a bit smaller so that I can move it a bit closer. Okay, so I guess this is your sarcoplasmic reticulum, not to scale, but sarcoplasmic reticulum. All right, now these calcium ions that come in, you might think that the calcium ions, these are the calcium ions that do the job of, binding to troponin. Oh, I forgot to label this. So, troponin. You might think that, but there is not enough, these aren't actually the ones that result in in most of the binding to troponin and tropomyosin moving and resulting in contraction. It's actually the gradient, in these skeletal muscle cells at least, it's the gradient, the voltage gradient, that causes the sarcoplasmic reticulum to release more calcium and that calcium is what does most of the binding to troponin. Because I don't want this video to be too long, I'll talk about cardiac muscle and smooth muscle and how they contract in the next video.